Hello, everyone. Welcome to the chapter on transcription. In this chapter, we will learn different steps of transcription, which will be broken into 3 steps: initiation, elongation, and termination. And in that process, we will also learn about several components involved, such as what is a promoter, what is a terminator, and the most important enzyme, RNA polymerase, and then some of the transcription factors. So we have to learn, we will learn the general concept to start with, and then we will learn specific details that are pertaining to prokaryotes and eukaryotes. But we'll take a quick review of what we have learned in the last class. I have discussed about ORF, what is start codon, some of the start codons, stop codons, genetic code, and so on. And also, I hope you remember reading frame. We will look at the reading frame in the next chapter as well. So there it will make much more sense. But in this case, I want you to remember this: the simple way of saying transcription is formation of, or it is DNA-dependent RNA polymerization. Okay, so DNA-dependent RNA polymerization. When we say that, that means DNA will act as a template, and then RNA will be synthesized. And why do we call it as transcription? Transcription means something like rewriting. Okay, it's rewriting the same thing in the same language. Here, the language is nucleic acids. Okay, so 1 second, something is wrong with my marker, but okay. So nucleic acid, the language is the same. It's a language of nucleic acid, except that it is copied from RNA to, from DNA to RNA. So you term it as transcription. Whereas translation, it is from the language of nucleic acids to the language of proteins, or we'll just use proteins. Right? It is a different language. That's why you can think of it as being called, that is the reason for being called as translation. So we also learned a few things, a few concepts or terminology. We learned about what is a template and what is a non-template. Template is the one that acts as the template for synthesis of RNA. And the polymerization, like in DNA polymerization, happens by base complementarity, by Watson-Crick base pairing. So the strand, the template, and the newly synthesized strand will always be complementary, which means that the sequence of this newly being synthesized strand is identical to the top strand here, right? And I also told you about RNA is mostly called the plus strand because it has the open reading frame. It is also called as the sense strand, right? So I hope coding strand, it is called the plus strand. It is also called the sense strand. And based on that, you can think the only difference will be about template and non-template. The strand that acted as template in transcription is called as the template. The other one, or the complementary strand, is called the non-template. But it is similar in sequence to that of mRNA. So coding strand, plus strand, and sense strand. So I hope you will try to catch up with those terminology wherever used. So the enzyme is RNA polymerase. RNA polymerase in prokaryotes and eukaryotes, there are differences. One of the differences you have to remember is that in prokaryotes you have only one RNA polymerase, whereas in eukaryotes you have 3 different RNA polymerases: RNA polymerase 1, 2, and 3. They have different functions, but we'll get to that later. RNA polymerase is a multi-protein complex. It has several subunits. Okay, if you see the eukaryotic ones, they have even more large number of subunits. Okay, so RNA polymerase has something, it is special in many ways. We will think of it in the next slides as we go. But right now, what I want you to remember, to try to figure out what are all the activities that it does. Say this is the DNA. Here is the DNA molecule. It has been unwound. And as the RNA polymerase is moving this way, you will have more and more positive supercoiling here. Okay, if we had a physical structure, we can always figure it out. What will happen, positive supercoiling or negative supercoiling? It will be positive supercoiling. So DNA polymerase, to start with, is unwinding the DNA. It is polymerizing the RNA, and it is then rewinding the DNA behind as it is passing along. And it is all these things. It has so many channels to do this, and also active centers that mediate each of these activities. So there is a template channel. You can think of there is the DNA for the incoming DNA channel. And then you have DNTPs, deoxy ribonucleotide triphosphates. Okay, nucleoside triphosphates, you can call them as. Sorry, 1 second. It should be, if this is RNA polymerase, this should be NTPs, ribonucleotide triphosphates, isn't it? And they come in and they get polymerized here in the active site. You have a DNA hybrid, the RNA DNA hybrid forming, and here the RNA is broken down. I mean, the hydrogen bonds between the hybrid are broken down, and the newly synthesized RNA is being pushed out of the active center, and then the template strand here will start base pairing with the non-template strand after the transcription, as the RNA polymerase is moving in this direction. Okay, so remember, here is, I think this is an error there. So DNTP, deoxyribonucleoside triphosphates, are required for DNA polymerization. The substrates for RNA will be nucleoside triphosphates, NTPs. Okay, and the area within which the DNA is denatured is called as the transcription bubble. That it is a transient thing where the two strands are denatured. So the chemistry of polymerization is very much similar to that of chemistry of DNA polymerization. If you see, there are two metal ions, like in DNA polymerization, we call them there metal ion A and metal ion B. And here also it is almost same, function is exactly the same. The magnesium ions will stabilize the beta and gamma phosphates. This would be the alpha phosphate. The other magnesium ion, along with some other residues that are here, will activate the 3' OH of this, and that oxyanion will make a nucleophilic attack and form a phosphodiester bond like this. And this will be broken off, and that will be, these two phosphates will be released as pyrophosphatase, which can further be broken down to release more energy. Okay? So it is, the chemistry is same. Yet we have so many differences with DNA polymerase. Let's try to have a comparison of DNA polymerase and RNA polymerase, so that we'll be able to appreciate, we'll be able to review what is DNA polymerase do, and also be able to appreciate how special is RNA polymerase. So in DNA polymerase, in both the cases, you have, in both the cases, DNA is the template. Right? In both DNA polymerization and RNA polymerase, both of them require DNA as template. The substrates are different. In DNA polymerase, the substrates are DNTP, deoxynucleoside triphosphates, whereas for RNA polymerase, they are RNTP. You don't have to write RNTP, but if you write, it is easier for everybody. So ribonucleoside triphosphate, ATP, GTP, CTP, and UTP. Whereas for DNA polymerase, you would have had DTTP, right? Uracil and thymine. Then there is, when we were learning about DNA polymerase, we have learned that DNA polymerase cannot initiate replication by itself. It needs a 3' OH, right? This is what is required. For that, during replication, we have an RNA primer synthesized by an enzyme called as primase. That primase is actually, technically speaking, it is an RNA polymerase. Only thing is that it is associated primarily with the replication. So we gave a special name and so on. But the RNA polymerase that we are discussing here is mostly involved in the transcription of everything else for cellular gene expression. The point here is that RNA polymerase, as such, does not require a primer. It has the capacity to synthesize RNA from anywhere. It can initiate by itself. Okay, all it needs is to make a stable binding with the DNA, and then create this transcription bubble, and from there transcription can initiate. Whereas DNA polymerase cannot do that. It has to have this 3' OH, and then the RNA polymerase will usually do the job for that.
Adios, amigo. Powering on.
Sorry. And then there is also fidelity issue. DNA polymerases have polymerase activity, and they also have 3' to 5' exonuclease activity. If you remember, some of the enzymes also have, some of the DNA polymerases, like DNA polymerase 1, also has 3' to 5' exonuclease activity. So this 3' to 5' exonuclease activity increases the fidelity. That means if there is a incorporation of a wrong base, then it can remove it and restart so that the wrongly incorporated one is eliminated from the DNA sequence. So DNA polymerases have a high degree of fidelity. That means accuracy of base pairing. When it comes to RNA polymerase, it's not so accurate. It makes about 1 in 10,000 bases of polymerized bases, one mistake per 10,000 bases. Whereas DNA polymerase could be 10 times more accurate, and some of the enzymes are even more accurate. I hope you also remember that when we are talking about DNA polymerases, we spoke about Pol 4 and Pol 5. I'm reminding you so that you will be able to recollect what you have learned, and if you couldn't, it's a good idea to go back and have a look at them. I find them highly inefficient, they are inaccurate, they are very slow, yet they are very important for bacterial survival. So it's a good idea to have a look at them and try to have a discussion with your own self. So RNA polymerase is special in some ways. There are common features that template is similar. There are differences in types of substrates, RNTPs. It is primer independent. It is not as accurate as DNA polymerase, but it is still good. Considering that. Or I have to ask a question. Think of the DNA is like this, okay? And RNA is also being formed. Where do you think fidelity is not so important? If fidelity is, or the polymerization rate is bad in DNA, then you are actually causing them mutations. That means majority of the mutations are detrimental, and that means that cell would die. But if you have a mutation in the RNA level, it's not a permanent thing. It's a temporary thing, because every RNA is made and it's also destroyed. That's not the case with DNA. DNA is the master copy. It has to be retained as it is for as long as possible. Okay, so those are some of the ways you can think of or for trying to reason why this is so. If DNA RNA polymerase was highly accurate, then its speed would be compromised. If its speed is compromised, then the number of transcripts made or the mRNA molecules made will be less. That means the cell is going to be slow. So this is a mitigation between what is absolutely required for survival and what is the best possible speed that we can have while we achieve the goal of survival and reproduction. Okay. Broadly, the steps of transcription will be 3 of those. First one is initiation, second is elongation, and the third is termination. So we will just have a brief look at it, and then we will go into the finer details. So RNA polymerase does not just bind anywhere in the DNA and start transcription. It starts in specific locations because those locations have high affinity for RNA polymerase to bind there. Like you remember when we were studying about DNA polymerization, we know we have studied that DNA polymerization initiates only at origin of replications, not randomly anywhere. They have to be coordinated things to happen. In the same way, with transcription, we also have certain locations within the DNA. Those are referred to as promoters. That is the place where RNA polymerase, there are other components also involved, but just that is where RNA polymerase binds and will start transcription of this downstream one. Always that is how it is put. And if you remember, usually a promoter, if you're drawing a line diagram, it is put as something like this. Say ORF will always be put like this, right? There is a arrowhead like here. That means this is an open reading frame. That means here you have the start codon, AUG, or at DNA level, I'll write ATG. And this arrowhead indicates the direction of reading the ORF. Okay? And at the end of here, you would have stop codon. This is about open reading frame. I mean, that means it actually, when ribosome reads it, it will produce a protein. But we are talking about transcription here. And to indicate that, they will put an arrow mark like this, a broken arrow like this, which means it is showing the direction of reading, right? This is the direction of transcription, and that is where it is going to start. So that is representing promoter. Promoter is usually represented like this. Okay? Anyway, so you need to understand why the arrow heads are like this and so on, so you'll be able to find out easily. The direction of transcription and the direction of the ORF should be in the same. If there is a situation where there is something like this, an open reading frame is read in this way, and the promoter is here, you're actually not going to get the sense strand. You will get the antisense strand. For a promoter, for an open reading frame oriented like this, again, promoter will be read in 5' to 3' direction, right? This is actually, it would be in the lower strand because this is 5', 3', 5', and 3'. So if it is like this, you should have had a promoter this way, so that you will produce the start codon till the stop codon, and that is how you will have. We will use some other class to discuss these things again, don't worry. And I hope you are all aware of the exam schedule that has come up. Please have a look at it. I think I already shared it to you. Anyway, so I'll just describe the transcription, what happens. Okay, I have to also describe this. What is here? You see +1. +1 is the first base added during transcription. Okay, that is where transcription actually has started. That is represented as +1. So if you think of it, you would have an open reading frame here. You would have ATG start codon here and TGA stop codon here. And this region, okay, this is called as 5' UTR. I think you remember when we were reading about transposons, we came across this acronym UTR. And this is on the 5' side. This is the 5' side. So you call it as 5' untranslated region. Okay, the mRNA, when it is produced, will be something like this. It will be this long from here till here. And the open reading frame is from here to here. So you will have, this is the 5' UTR, and this part will be the 3' UTR, untranslated region. Okay, that's good enough with the terminology stuff. So once the RNA polymerase binds to the promoter region, it makes, there are two complexes, closed complex and open complex. The difference between these two is whether melting happened or not. Melting here is transient denaturation of or a small stretch of denaturation. So here there is no denaturation yet, so you call it as closed complex. And once denaturation has occurred, a small stretch of DNA, then you call it as open complex. And then there are certain regions of RNA polymerase will try to move like this, and it will produce small stretches of RNA polymerase. Okay, we will discuss about this. I hope you have seen the videos. If not, have a look at it today, and then you will understand it better. So RNA polymerase will start synthesizing RNA from here, and as slowly RNA polymerase will keep moving in this direction like this. And as it is going, this transcription bubble will keep on moving as well. That means there are breaking of hydrogen bonds as it is moving, and behind it, there are remaking of the hydrogen bonds within the template DNA. I mean, template and non-template DNA. So that is how, that part will be the elongation, where RNA polymerase will keep moving and produce RNA and as it is moving along. At one point, it will stop. That stop, there are certain regions within DNA which are downstream of the promoters after a long distance. Those are called as terminators. When those sites are reached, RNA polymerase will cease to synthesize any more RNA and will dissociate from the DNA. And that part is called as termination. So if you see, there are, as a summary of this particular, this slide or the overview of transcription, you can see it as initiation, binding of RNA polymerase, formation of open complex, small stretch of RNA is produced. Elongation is just keep on going until you reach a terminator. Termination is when the terminator is reached, RNA is dissociated, newly formed, RNA polymerase is dissociated from the DNA as well. So we'll first look at several components that we have discussed. I think today's focus will be mostly about the different types of the molecular aspects of initiation, because that has many more things. Elongation and termination are a little simpler. So there are several types of promoters within bacteria, and what we are studying now is about bacterial promoters. It is well understood and best understood. Eukaryotic transcription is a little more complex. We will get to that anyway. So typically, we have three different types. One of it is the plus one, I hope you remember, and the broken arrow, which is kind of indicating where the promoter is, and the plus one is where you have is the first base that is added during transcription. Typically, we have a region called -10. That is, -10 means from here you have to count one base here, it will be -1, -2, -3, -4, like that you keep coming, then you'll have -10. And there is another conserved sequence which is called as -35 sequence. And some of them do have a region that is called as up element. These are all DNA sequences. This is whole DNA. These are some of the important DNA sequences that are required for the binding of or loading of RNA polymerase onto the promoters. So up is for kind of upstream element. This is when you're reading about transcription, anything about this here, these are all upstream sequences. The ones down, these are all called as the downstream sequences. So because it's on the upstream, upstream of -35 and an additional component, you call it as upstream up element. In some cases, there you have -10, and it's called as extended -10 because -35 region is not there, neither is up element. What you have is an extended, some more region, some more bases are there, which are important for the binding of RNA polymerase. So this is called as extended -10 type. There is one more, which is called as, which has -10 and -35 regions, but it is also has a small stretch of bases, which is called as discriminator. And those are some of the types of DNA, promoters that are present in bacteria. So when I said -10, -35, it doesn't make so much of sense. What I mean to say here is, if you see the bases, I want you to focus on what is meant by consensus. Okay? Let's see. I hope I'll be able to make it up. Let me say I will write a T, G, A, C. I'm just making a random sequence. Don't be alarmed about it. This is sequence 1, sequence 2. I will write about 4 sequences. Otherwise, it will become too much. So CAT and GTCT. So I'm just trying to build what is a consensus sequence from what is available. So let me try if it works out. So it's like doing an addition or subtraction. You can think of it like that. In position 1, you have all of them have C. In this case, it's not conserved. A in some cases, T in some cases, right? In other ones, in third one, fourth, fifth, it is CAT. And in the next one, it is not. And in that also is not. And then you have CT. The sequence here, CAT, C, and CT, you can call them as consensus. That means you take different promoters from different genes of one organism. Say you collect all the, from E. coli, you take all the promoters and then put them in an order like this. And then see what is there, what are those bases that are highly conserved. If you see that in -10, the consensus is TAT, AAT. What does that mean? That means in majority of the in majority of the E. coli promoters, in the -10 region, the sequence is TAT, AAT. It doesn't mean that there would not be any variations to it. Say, for example, with triptopiron, which we will study in the last unit, it has a C here, there is a difference here, TTA, CT. What is the consensus? It is TAT, AAT. Similarly, in lacoperon also you have, it is very much similar to TAT, AAT, but there are also slight differences. So you have, one can remember -10, the consensus sequence at -10 is TAT, AAT. Similarly, if you look at -35 sequence, you will find that TTG, ACA. This is the consensus for -35 sequence, excuse me. Okay. And here N6 is put, N17, meaning that the distance between +1 and here is about 6 bases, approximately. It can be 1 or 2 up and down as well. Like you can see here, it is the difference spacing between this is about N8. Even though there is little bit change, people still call it as -10 region. Okay, when they first identified, they found that it is -10 region has high consensus. Only later, slowly more and more genes and promoters were identified. And then we see that there are little bit differences in the spaces. The count may vary a little bit, but still following convention, we call it as -10. The same is the case with -35. Here you have N17, meaning that the distance between -10 and -35 is about 17 bases. There could be alterations here, say N16 and N18. That is the spacing between these elements. So why are these conserved? Which would mean, if they are conserved, they are important, right? Important for what? Important for transcription, yes. How are they important for transcription? They are important for transcription by making interactions with the RNA polymerase like this, right? These are conserved regions because the RNA polymerase, or through some other factors like sigma and so on, the proteins have a way of recognizing these particular sequences. They do not care what sequence is here. What may matter, though, is the size of this spacer. So the sequence is not a problem in this case. Size is important. Because if they're two apart, -35 sequence is here and -10 is somewhere, and the protein is like this, right? It cannot interact simultaneously with -35 and -10 if they're far apart. I hope you got some understanding of what I meant. There is also up element. It's not always in all of them. A few of them have, or some of them have. So there is, the RNA polymerase also has carboxylic terminal domains which interact with the up elements. Okay, we'll get to that in a few minutes. I think in the next slide it is there. But I hope you understood what is meant by consensus. Consensus is those conserved bases are trying to determine what is conserved within this region, the sequence. Those are called as consensus. That means they are conserved in several promoters. -10, so +1 is the first base added, and it is usually the A. It is in the +1 place, you usually have adenine. Then about 6 bases spacing, you have -10. The sequence is TAT, AAT. Then there is a spacer of about 17 bases. Then we have a sequence called TTG, ACA. And there are up elements. We don't have to mark up all these things. Anywhere you find N, which means any of the bases, nucleotide, any of the nucleotides. If they put A and T like this, that means it in that position, either it is A or it is T. Okay? If it is written A like this, it means only adenine is present in that position. So while we say that these are the consensus sequences, there can also be differences within these sequences, very slight differences. Okay. So this is, I hope you will be able to understand what I meant. I have drawn a sequence for determining consensus, and this is nucleotide frequency. So here you have, this graph is the same thing, except that it is shown in a tabular form. So -10, I will only discuss these two, okay, so that we'll move on. In this position, okay, you can see in how many bases, in how many promoters, which base is present. This, which would mean that about 75% of the promoters have T in this position. That's a very huge conservation. In the second position in the -10 region, if you see like this, you can see that some of them, about 60% of them or so, have A. About 10 or 15 or 20% have, there should be a key somewhere. Yeah, this will be C. Okay, I'm correlating with this C here. C, and then this must be G, and this is T. Okay, you'll see that in more than 50% of the promoters, A is present in the second position of -10 region, and very little bit for the rest of the three. Okay, so you can read the textbook, and this is I picked this up from the box. You can read it, and you will. I hope you will be able to understand. If not, please get back. In prokaryotic transcription, we need something. RNA polymerase cannot just go and bind directly. There is an important factor that is called a sigma factor. The primary function of it is what? Is that it mediates the interactions between the DNA or the promoter, I should say, between the promoter and with that of the RNA polymerase. So it is something like sigma factor is what binds here like this. Okay, it interacts with the -10 and -35 region. Sometimes it may make interactions with the extended region. We have seen two other types of promoters, right? With extended -10 region. In some cases, we have seen about discriminator region. So sigma factor is important for binding the -10, -35 region, to which RNA polymerase binds, right? So sigma factor has a very important function, and that is to enhance or mediate the interaction between the promoters and the RNA polymerases. I think in gene regulation, when it comes, we will discuss about what is meant by this. There are many different types of sigma factors, not just sigma factors. There are many types of sigma factors. We will get to that after we finish the transcription, and hopefully, or in the regulation chapter, we will discuss about it. Right now, what we want to read is how transcription happens. Then we will go to later part. Yeah, so sigma factor will bind, and what is shown here is the -10 region, which is going to be interacting with the sigma 2 part. It's a part of the sigma factor. Just for understanding, you can put. So when it binds, it can remain as double-stranded DNA if it is like this. But if this A fits into this pocket and this T fits into this pocket like this, so you're breaking some interactions here, and then that will increase the probability of denaturation. So that will result in melting of the promoter. What is melting? It is the breaking of hydrogen bonds at the promoter region. So you can think of this as a part. DNA polymerase, all this part shown upwards is the RNA polymerase. It has alpha CTD. It's the alpha subunit carboxylic terminal domain. That is what interacts with the upstream element or up element. Here you have alpha N-terminal domain. I hope you remember that we read 5' to 3' DNA or RNA. When it comes to proteins, we talk in terms of N-terminus and C-terminus. N is for amino group, C is for carboxylic group. So this is the alpha CTD would mean that it is the C-terminus of the, there is a domain on the C-terminus, and this is the C-terminal domain of alpha subunit of RNA polymerase. Anyway, this part that I'm showing is the sigma factor. You have several regions designated as sigma 2 and sigma 4. There will be 1 and 2, 3 obviously. So sigma 2 interacts with the -10 region. Sigma 4 interacts with the -35 region. The alpha C-terminal domain of RNA polymerase will interact with the upstream element. Okay, this would be this is the promoter or transcriptional start site. You call it as +1. So once it binds, we also, when we are discussing about transcription, we said for transcriptional initiation, binding of sigma factor and RNA polymerase, then we have a closed complex, and then we would have an open complex, right? So the open complex, the formation of this open complex is about denaturation between -10 to +3 regions. Say, for example, if you had, this is the +1, so you can assume that +1 is here. So +3 would be somewhere here until -11 bases. So this much, or about 15 bases or so of DNA is DNA melts. And it is not an active process, meaning that it does not require energy. It's just an isomerization, which means to change its shape. Okay? Once this conformational change occurs, then it is open complex, and the interaction is irreversible. So I'll just go back again. So RNA polymerase has bound here, and it is in closed complex, right? Then there is also a possibility. So this is RNA polymerase and the promoter. They can form a closed complex like this. It is reversible, meaning to say that it can dissociate. RNA polymerase can dissociate from the promoter. But once within this region, when it formed a closed complex, and it also formed an open complex, and this is the RNA polymerase, that interaction is irreversible, meaning that RNA polymerase will not go like this, will not dissociate from that promoter at that point. It will have to go through transcription. Okay? So there are 5 channels. We already described it, but let's have a look at it. It is, they call it as pincers. It's like a claw of a crab. If you look at a crab, you might have known that it has some things like this, right? And it would pinch us. So it is there. It is an indication or depicting something like that. RNA polymerase will move in this direction, and it will keep on moving. And in this channel, we will just look at some of the channels. First is NT channel. That is where the nucleotides will come. T channel, that is the template region, the DNA template that is moving here. And you have an exit channel. This is the exit channel, that is where the RNA exit channel. And you'll also have the DNA that is exiting. How many are there? Yeah, we have NTP uptake, we have RNA exit, DNA entry, and DNA exit. And yeah, sorry, this is non-template channel, and this is template channel. Okay, what is non-template? It is not going to act as a template for DNA polymerization. If you see this region, that is the template DNA, which will be transcribed and RNA polymerase will form. RNA will form. The green ones you see here, Q3, Q4, those are parts of sigma factor, not Q, sigma 4 and sigma 3. Okay, if you see that the sigma 3, 4, this linker that is present, let me try again. If you see this linker that is present here, it is in the path of RNA exit channel. Okay, just keep in mind so that we will remember it. We will get to it in the next slide. So all the polymerization, there is something called as abortive initiation. I hope I will take this and then I will stop. Please watch the animations. It will give you a good idea. So abortive initiation is something like this, as the name suggests. So closed complex, and from there we get the open complex. Okay, you should assume that sigma factor and RNA polymerase are also associated. And when it is the abortive initiation in the sense, so once this is open complex, RNA polymerase will synthesize a small bit, and the bubble will move, the transcription bubble will now move to a different place, like this. A small bit, not too far. And the transcription RNA will be produced a little more bit, and then it stops. It dissociates. RNA polymerase falls off after making only a small fragment of RNA. It may happen, it may go through these steps several times, and that is called as abortive initiation. Typically, what should happen is that the RNA polymerase should synthesize a long stretch of RNA that is going into or moving into the elongation state. From initiation, it does not go to elongation state, instead dissociates. And it might happen several times in this location. And that is called as abortive initiation. Okay? Okay, this part, it is about elongation. We will discuss in the next class. But what I want you to know is elongation has proofreading, like we have seen in DNA polymerase. If you remember more, of DNA polymerization or replication, you can get more than 70% of transcription as well. Okay. So we will look up in the next class. We will look at some of the editing mechanisms, phosphorylated editing, hydrolytic editing, and so on. And then we will finish about. Okay, I thought I'll finish till here, but we'll do that later. And then we'll talk about termination and eukaryotic transcription in the next class. In today's class, we have learned about the overview of transcription, and we have seen the RNA polymerase types. We have learned about the chemistry of RNA polymerization, and we have made comparisons between DNA polymerase and RNA polymerase, and overview of transcription, and we have learned about different types of promoters. If you are planning to work in biology, please study this stuff very well. If you want to proceed in the future, if you want to research in biology, it's always important that you study these things properly and understand the textbook well enough, so that in the future, when you are supposed to read research papers, you will feel very comfortable reading these things. Okay? And I hope you understood consensus and the sigma factor and so on. Okay, I just want to use 2 minutes to introduce about the exam. The final exam I'm talking about. I will stop the recording now.